like I am, it's should MRD or minimal residual disease status be used as a good endpoint for drug approval? Now, minimal residual disease results come from an extremely sensitive test that detects how many myeloma cells are left in your body after reaching normally a complete response, but sometimes a very good partial response from myeloma medications. And some researchers are now wanting to use these MRD results as endpoints in clinical trials in order to accelerate drug approval. Now, this isn't the only way that researchers are wanting or seeing MRD results as being useful in the myeloma field, but this is what we're going to be talking about today. It may not surprise you that different researchers and different specialists have varying opinions on the subject. And so today we're going to be listening to Dr. Ali and what his personal opinion is on this topic, and how he is using MRD as well in the clinic. We're going to be able to ask him our questions, and I'm just really looking forward to it today. So Dr. Abbas Ali is a medical oncology, my words are kind of tongue twisting today. He's a medical oncology specialist in Baltimore, and he cares for patients with cancer. He earned his MBBS at the Aga Khan University, and he completed his residency at the University of Arkansas for Medical Sciences and performed a fellowship in medical oncology at the National Institutes of Health. Dr. Ali is an amazing physician, and I'm really excited to hear your opinion, and thank you for your preparation for this, and I'm looking forward to what you have to say.
Yeah, hi. Thank you so much, Audrey, and to HealthTree and to everyone that's listening. Thank you for giving me the opportunity to speak. We're going to be talking a little bit today about MRD in myeloma, a little bit about the MRD, a little bit about my thoughts about the MRD. And bear with me here. Can you see
Not
these
yet.
slides?
Not yet. Mm-mm.
Do you see?
I just see you. So if you want to go back to that main Zoom screen.
Yeah, bear with me here. The screen is shared, so okay. Is this working?
Yes, now it is. Mm-hmm.
Okay. Yeah, so we'll be talking a little bit about MRD. These are my disclosures. I don't have any relevant disclosures. And I think the relevant disclosure that is important that I want to start with is that I do use MRD. Or rather, that's not quite accurate. I do get MRD and I obtain it often. And I am a fan. And I'm a fan of learning more about it and what directions it will take us in and how it might potentially help our patients. And so we'll go over some of these thoughts through this talk. We'll talk a little bit about what MRD is, what the testing is, and what the implications are for having MRD positivity and what does that really mean. Whether what MRD looks like in clinical trials, right, for drugs that many myeloma patients may be on and clinical trials that many myeloma patients may have seen. And then just some of my thoughts on its relevance for drug approvals and clinical trials going forward, and thoughts on addressing the MRD positive space. So and is this stuck in? No. So when I think about myeloma in a newly diagnosed space, and I think that, and I presented these slides before, our Arkansas experience when I was down in Arkansas at the Myeloma Institute, as well as the experience of many others, taught us that, myeloma therapy, more often than not, because the majority of patients on a long enough timeline will relapse, should be a balance and a combination of intense and continuous. So generally people get induction, then they go on to a transplant when eligible, and then a maintenance strategy. And over time, what we have seen with the survival curves versus, say, the year 2000, where things are very different versus where they are now, is that survivals have thankfully improved remarkably well. And that is because of the way the newer drugs are utilized. So continuous suppressive therapy, factoring in transplant, using good drugs, having a maintenance strategy. So it's the intensity and having an armamentarium of drugs that have really changed things. And so really what we all want to do is see what we can do to continue to extend these responses and extend these remissions. And when I showed this slide at an earlier point in time, I had thoughts in mind about, using intensive therapy like quadruplets, do a transplant, maintenance strategy. But I was thinking a bit more about depth of response in the MRD context. And we hear more and more, and I'm sure our patients hear that, look, everywhere we go, they talk about this MRD and the implications of MRD and how everything is now reduced down to MRD. And I guess my question here is that, does everyone really need the deepest response all of the time? Because on average, we do have data. In general, we do have data from trials that show, yeah, MRD negativity, deep responses are good. But is that always applicable to every patient? And what contexts are there that where that might not necessarily be the case? And where there might be other strategies that don't necessarily involve intensive treatment to get to an MRD negative state continuously, every last little myeloma cell. So what we see is that over time, the depth of response, PFS, and overall survival and MRD have increased and improved as drugs have developed through the early 2000s and through the latter part, the more recent part of the 21st century. And that depth of response, it's very clear that when we look at data, and these are published by our colleagues, Prashant Kapoor and colleagues at the Mayo Clinic, that over time, what we're seeing is that with the new drugs, with the incorporation of transplant, the progression-free survival, or rather progression-free duration, and the chances of staying that way over time remains higher the deeper the response you get. And so, yeah, that's a straight up thing that, look, maybe it makes sense that we can push the response deeper as much as possible. And when you take complete remission, right? And so if you take this blue line, which is complete remission, and on the y-axis, what you're seeing is that the chances of remaining progression-free, which is that the myeloma is controlled over time, is higher if the bar is higher. And so these are patients who are in complete remission. So it says that their chances of being in remission or myeloma control over many years is higher if you're in complete remission. You take it over to the graph on the right, and it remains true when you go deeper and say that, look, if we look at near complete remission, which is now known as a VGPR, or a complete remission, and then on top of that, compare that to a stringent complete remission, which is deeper still, then if anything, outcomes are even better. So you know what, why not just keep taking this to its logical conclusion? Let's just go as deep as we can get. And so in this very nice meta-analysis that was initially published by Nikhil Munshi and colleagues in JAMA Oncology in 2017, and then there were follow-up studies as well that they've shown numerous. They looked at this test that people have heard of and are here to hear about, which is MRD. And they're like, look, if we take a very deep look at what's in the bone marrow and take our more sensitive tests, does that really make a difference from what we have traditionally seen? And you see that looking at the same survival figures of progression-free survivability, which is really should be called progression-free duration, and indicating that myeloma is remaining under control, as well as overall cumulative survival, there's a difference between the two lines where the MRD negativity is high, line is higher over time versus MRD positivity. Okay, well, let's delve into this a bit deeper, right? Is that still hold true when we look at that test, when we're just looking at patients who have achieved a complete remission? Because traditionally, up until this point in time, or until very recently, complete remission or stringent complete remission seem to be enough. Complete remission is very well defined, and there are multiple trials looking at complete remission in a randomized fashion as an endpoint. There are few trials, or very, very few rather, no real trials that really look at stringent complete remission as an endpoint into itself. But for our purposes here, complete remission is good. And we see the same thing, which is that, look, getting to a deeper remission than when you are, than than what we traditionally consider to be a complete remission is better. But what does response for myeloma really mean conceptually? This very busy looking slide is a summation, and I put it this way without simplifying it to indicate how dense the thought process is behind the depth of response in myeloma. And when you talk about the International Myeloma Working Group response criteria, they're pretty detailed. But conceptually, I like to try and make it simple. I say that, look, responses in my mind are, let's say, minimal responses where the numbers don't come down by a lot. Partial responses where the numbers come down by 50-ish%. Better than that, 90-ish%, very good partial response. This is really looking at markers in the serum. And then moving on to what's a complete remission and the stringent complete remission, immunophenotypic complete remission, and molecular complete remission. But patients often ask, all right, so I have well-controlled myeloma. Am I in a remission? But remission in myeloma is different than it is in a lot of diseases. In lung cancer or colon cancer, you do a scan, you don't see any disease, well, you're in remission. The same thing doesn't necessarily apply to myeloma, which is interesting in that it is a hematologic malignancy that in some ways behaves like a solid tumor. So when we take all of these definitions, what they're really telling us is that we're reducing tumor burden beyond a point where it's traditionally measurable. And now we're trying to look at the very tail end of that disease, the last few cells, wherever they may be hiding, and trying to figure out, can we use this to make decisions? There are two main ways to do this. When you guys look at your bone marrow biopsy reports, or you look at any other reports and they talk about this flow cytometry, basically what that means is that they are taking cells from the bone marrow and they use antibodies which light up to tag them. And they run all these cells through a device where a laser shines through, and you get this nice pattern, which is why it's called the multicolor flow pattern, which is interesting, but not necessarily good wall art. But this gives us a sense for the molecules on the surface of the cells. What this test is doing is trying to look at the molecules on the surface of these cells and decide, hey, are there plasma cells or myeloma cells that are in there? So flow cytometry is a very good tool. The modified version of this is what we call next generation flow cytometry, which is what we use in the measurement of minimal residual disease by flow cytometry. When you look at what's called next generation sequencing, which is the test that I like to use, that I'm a fan of, we find that what this test does is that it looks at the DNA sequence. It is looking for bits and pieces of the DNA of myeloma cells inside the bone marrow. And how it's doing that is by looking at this VDJ sequence, which is what's next generation about it. And the VDJ sequence is specific to lymphocytes that turn into myeloma cells. So myeloma cells have this sequence and they retain this sequence or a slight variation of it throughout the course of the myeloma. And so it's a useful tool to say that, look, if I can identify this sequence, I can continue to look for it by a very sensitive test. And what they can do is when they perform this next generation sequencing, because it's a very sensitive test, when they get a bone marrow sample, they sample millions of cells, ideally more than a couple of million, and try and see that, hey, are there any myeloma? Is there any myeloma DNA in there based on this sequence that has been previously identified? How is this sequence identified? There's an FDA-approved test by this company called Adaptive out in Seattle called the clonoSEQ. And what clonoSEQ does is that when you run or you ask them to run the test, they identify a very early sample, or the initial sample rather, of the myeloma in the bone marrow. And then they study what DNA sequences might be in there. And then when you go back to them and say, hey, I have a situation where we've done our treatment, maybe we've done a transplant, the myeloma seems to be under excellent control, we're in complete remission. I'd like to see if there's any evidence of myeloma left over. They go back and look for those sequences and see if they can track them going forward. There have been other mechanisms of looking at MRD, those have kind of fallen by the wayside, but it's basically come down to these two. And they have comparable sensitivities. What does that mean? That means that if you go to the next generation sequencing test, if it looks at a million cells from the bone marrow and there is one myeloma cell in there, it will find. That's how sensitive it is. When you look at the next generation flow cytometry test, then it can also get down to a situation where if it looks at a million cells and there's one myeloma cell in there, it can identify it. But that's a little less consistent than the consistent ability of next-gen sequencing, in my mind, to identify a myeloma cell down to a very high degree, one in a million. Next generation flow cytometry can get to 10 to the minus 6, which is 1 in a million, but is often reported as 1 in 100,000. Also, the advantage with the adaptive clonoSEQ, which is FDA approved, is that it's a reproducible test that you can have standardized across institutions, across hospitals, across testing platforms. Next generation flow cytometry, there is a standardized test that they use in Europe called the Euroflow, which, and we have comparable tests here, for instance, at our institution. But the tests in different labs are all slightly different from each other, and it becomes a little bit difficult to compare. One of the advantages, though, of that next generation flow is that, yeah, well, if you have a lab that can do it, you can do it in-house, and you can turn it around quickly. With next generation sequencing with adaptive, you can certainly do the test, but it's required to send it off, get a sample, send it out to a company. It takes a little bit longer for it to come back. To keep things even and to be able to compare results going forward, typically what we have done and what I have done is that on my patients where I'm getting MRD, I get clonoSEQ by adaptive so that everything is comparable. And I have a sense for what each time point in the course of, say, MRD tracking for one patient means, because I have the same test all the way through and I can compare the two together. And the way they obtain these is by bone marrow biopsy. They can do this on the marrow itself, but usually they like the aspirate. What the IMWG criteria then say based on MRD is that, look, there are different ways to interpret this MRD. And you can, what they like to see is a sustained MRD negative, whether you're using next generation flow. So if you get MRD at serial time points, they want to see that it remains negative, and being in a sustained MRD negative situation is a positive situation to be in. And it also illustrates the having sustained MRD negativity as a criteria here in the International Myeloma Working Group criteria and the NCCN kind of illustrates that a sequential time points or sustained trends over time matter more than single time points. And this is a conversation that my patients and I have often when numbers change or MRD changes. It comes or it goes, or it's higher, or it's lower. Before we overinterpret blips or results, let's look at what the trends may be. So having trend lines is important. You can be flow MRD negative, sequencing MRD negative, which is when you've done NGS and you don't really find any myeloma. But then there's this imaging part of it. And that's very, very important because human beings are patchy. Our marrows are patchy, our bones are patchy, everything is patchy about us. And when we are doing a bone marrow biopsy and getting a sample, we want to be able to put it in context. We want to be able to say that, hey, if I'm going to say that there's this true MRD negativity, I want to have complementary imaging. I want to have labs that show that my numbers are actually under control. There are more nuances to that, though, because what we're looking at is proteins. And proteins, especially when they get really small, can linger in the blood for a while, a good long time. Sometimes the proteins are there because there is no active myeloma as such, but there are, call it MGUS variants or other plasma cell variants that are there, and that may obfuscate the picture. So having a thorough context with marrow labs and imaging in particular is really very important. And it goes deeper than that. When you look at the Arkansas group, what they will in these large analyses looking at multiple patients, what they found is that getting imaging is important to understand MRD, because if you have a bone marrow which is MRD negative, but imaging somewhere shows that there's a little bit of something involved, then outcomes are a bit different, more in the middle. So this line shows us that there are some patients that might be MRD negative by MRD negative testing, but because the imaging was suggestive, then the outcomes are somewhere in between. So it changes things. For me, though, specifically what it comes down to is that now that I've got MRD, as the song goes, now that I have MRD, what do I do with it? When we first started doing that, this song was the first thing that went through my head is that I'm going to do this. I think we need to do this. but how am I going to use it to make decisions? How am I going to grow our ability to understand what MRD represents? And there are a lot of studies that have kind of helped us get there, that MRD has relevance in many different settings. This is the IFM DFCI 2009 study. This is an important study. This is one of the primary reasons we are still doing auto transplant. This is the French arm of the study. There is a US arm called the Determination Trial, which many may have heard of. It uses a triplet combination VRD. Nowadays, for induction, more people get Dara VRD. Previously, everyone used to get VRD. And what the study showed was that, look, if you look at patients who get transplant or who get VRD, then patients get into a complete remission, maybe higher in the transplant group. But by the time you get to MRD negativity, what you see is that there's a higher proportion of people in the transplant group who are MRD negative. So this is a question that comes up that, doc, you know what, if I am MRD positive, will a transplant potentially help push that response deeper? Well, that's true to some extent. You see, transplant does increase the rate of getting to an MRD negative state. And over time, though, with transplant, what we see is that even if you weren't necessarily MRD negative to begin with, or MRD negative after, over time, those responses can deepen. And for patients who are follow their own responses very, very closely, what I try and tell them is that we're not in a sprint, we're in a marathon, and this is something that we have to kind of follow over time, and that this is something that we have to follow dynamically. When you take this further, then it said the transplant part of it from this study said the same thing. Look, if you're MRD negative when you're doing a transplant, then that's generally a better situation than being MRD positive, whether you're looking at progression-free duration or long-term outcomes. And sensitivity matters because MRD negativity is a good place to be for most people, especially if it's down to a deep level. But what's interesting when you look at this line right here is that these are MRD negative patients on this study that got there, whether they got a transplant or they did not. So with or without a transplant, maybe MRD negativity for a lot of people is the same. And I think that's certainly true in some situations, in particular for patients that have more standard risk disease. But I think there are a lot of nuances to that, and also a lot of nuances to what the case might be in patients who have more high-risk features. And I think there are more recent trials that sort of help educate us as to that. When you really look at other trials as well that don't necessarily involve transplant, then you kind of see the same thing, that having deeper remissions, these are with daratumumab combinations. All these trials that I mentioned here, Pollux, Castor, Alcyon, Maya, these are all trials that involve daratumumab triplets versus other doublets. And they're showing that, look, being in a deeper remission, even with these treatments, when you may not necessarily be doing a transplant, is better and better for long-term outcomes. Let's take it to the next step. The master trial, which was recently updated, is a very nice study that Luciano Costa presented. This was a MRD adapted study, right? Because right now, so far, what we've been doing in the majority of cases is kind of having MRD as a test that's built into these clinical trials. But this particular trial was very nice, and there are others sort of in the same vein that are now sort of ongoing. And what it said was, look, we'll give people intensive therapy with the induction of 4 drugs, and then we'll do a transplant. And at each point, we'll kind of look at MRD negativity. And if you're MRD negative, we call that MRD sure, you might get a treatment-free observation at these different time points. And this was really an excellent study. And Luciano updated these results at the EHA meeting in Europe recently. But there's a few things that we learned, which is kind of illustrated here on the right. Yes, being MRD negative is a better place to be. But we also learned that MRD negativity, to some extent, especially in patients where it says here in the extreme right, have high-risk disease, MRD negativity in the very high-risk setting has different relevance than it might in the standard risk setting. Patients who were MRD negative in the high-risk settings did not derive as much benefit as patients who might have been standard risk. We kind of also know this in the practical setting. Many patients who may be listening to this follow what happens in the world of CAR T cell therapy. And we find the CAR T cells for myeloma since the very first CAR T cell trial at the NIH, that Abecma and Carvykti CAR T cells for myeloma have shown very deep responses. But as opposed to some patients with lymphoma or leukemia, for instance, you are not necessarily finding that we've hit the cure that we had all wanted to hit. We're finding that even patients, and the bulk of these trials so far have been in the heavily relapsed space, that a lot of these patients who have heavily relapsed disease, which might be aggressive disease, even if they become MRD negative, still relapse. So there's more for us to learn about that. And there's more for us to learn about what MRD negativity means, especially in the high-risk setting. But I'm going to take this just a little bit further with a quick case. I have a patient. He's a 63-year-old gentleman who was initially diagnosed with myeloma in 2013, and he was abroad. He was outside the States when this happened, and he had x-rays and he had a lot of lytic lesions. His IgG was pretty high. I don't have results, a lot of the results that were labs that may have been done when he was out of the country. And so we don't really know if he had lambda light chains or kappa light chains of diagnosis, because those labs and those tests in the bone marrow were never really done. He got Velcade and thalidomide and dexamethasone between 2013 for 5 to 6 months, and then he was on thalidomide maintenance for less than a year. And I've been following him for a number of years, and every time he comes here, this is December 2017, the immunofixation, which is a test that identifies whether there's a clonal protein in the blood, shows no MGUS or no myeloma. But then March of 2018, he has some kappa-free light chains that are seen on the immunofixation and some lambda-free light chains. Very confusing. In November 2018, it's just lambda light chains. June of 2019, everything looks clear. In between, in 2020, he had tests that were intermittently bouncing around between negative and positive. But in January of 2021, he's lambda light chains. And in August of 2022, there's nothing. And then again, recently, there's lambda-free light chains. So I haven't performed an MRD test. And I want to clarify that this gentleman is off treatment for many, many years now. But if you have a monoclonal light chain that you can find on immunofixation, technically that is enough. That is an MRD positive patient. If I were to do an MRD test, whether by next generation flow or next generation sequencing, I should find minimal residual disease. But should I really go upsetting that apple cart? Does this person have myeloma each time that the light chain seems to come back? Then should we run and get a ton of additional tests? And because we're concerned that the myeloma is immediately coming back. There are data to show that patients that were MRD negative that become MRD positive, and that if the trends continue over time across various trials, that does have implications towards relapse in the future. But I don't think that everyone with a teensy-weensy bit of a light chain, perhaps, or a teensy-weensy bit of plasma cells or myeloma cells that we find is necessarily somebody who might relapse. And this is something that we have to kind of identify and delve into a bit further with additional trials. And this is a source of anxiety for a lot of patients who are like, is my disease controlled? Is it not controlled? Does it need to get deeper? Do I need more intensive treatment? For patients that are looking at this, they're wondering, why has this person put on this picture of what looks like the Florida Panhandle? And really what this study is saying is that when you look at myeloma cells in patients that were treated, and these are patients that were MRD positive as well, what you're finding is that there are some myeloma cells that kind of look like an MGUS, right, which is the precursor to myeloma, that can persist for decades and never do anything. And there are others that are not MGUS-like. They're kind of more like multiple myeloma. They seem to have a more aggressive disease biology. And again, this is in patients who were treated for myeloma who are MRD positive. And so when you look at patients who get knocked into an MGUS sort of state, you see that the proportion of people who are in remission over time is, or who have not progressed over time, is higher versus patients who have plasma cells or myeloma cells that behave like real myeloma cells. So when I see that there is this big push by a lot of groups here in the States and in Europe towards, look, MRD negativity is the be all and end all for everyone all of the time, then I think, yeah, there are a lot of data that show that being in an MRD negative state is great, but I don't know if one size fits all of the time. And so what we need to do is find out where MRD is really applicable, where is a small amount of protein or a small amount of plasma cells really relevant? Let's go a bit further. There's more that goes into the control of myeloma and the prognosis, because people look at, hey, I've had my transplant, it's been months, maybe it's been a year, and I'm still MRD positive. And that's clearly very bad. But I don't know if that's always true, because there's more to controlling your myeloma and outcomes in a long run. One of these things that we're showing here in the study, which was again by Bruno Paiva, is that when you look at the immune profiles of different samples of myeloma patients, including patients who are MRD positive, then immune profiles that are enriched for mature B cells and other favorable immune cells have outcomes that are actually better. Yet all of these patients were still MRD positive. So there are a lot of things that go into deciding what the implications MRD may or may not have for the long run, because there are disease intrinsic factors, risk factors, what the fish is, whether it's a standard risk, high risk, what's the disease burden like? Is there extramedullary disease? What was the progression like? Was there a situation where getting into any remission or getting any response was challenging? Have relapses been very brisk? All of these things matter. There are patient intrinsic factors. Some patients are in excellent shape. Some patients are more like myself, not in the best shape. And all of these kind of have implications for, hey, what do my labs and a little bit of MRD positivity or otherwise mean? And there are also treatment-related factors, right? Different drugs have over time in various different trials shown us different efficacies. I like to tell my patients that, look, we shouldn't be looking at this in a snapshot, and we certainly should be over-interpreting figures, especially when they're fairly small. Blips don't matter to me as much. What matters to me are sustained trends in particular directions. What's also interesting is that when you look at immune environments and following transplant and following CAR T cells, we kind of see the same thing. A lot of responses. We were talking earlier about how there are still MRD negative patients with following CAR T cells that relapse, but favorable responses are seen in patients who have received CAR T cells, for instance, and have an immune profile in the which has more robust immune cells versus more robust myeloma cells, which is here on the higher side, on the upper panel. And if you take this a bit further and you look at this very nice and large analysis, a bit dense, might give one a headache, from the Tourmaline study looking at ixazomib, which is a weekly proteasome inhibitor in the frontline setting, we find some things that are very interesting. One of the things that's very interesting is that, yeah, we expected that patients that were MRD negative would have a slightly different outcome given periods of time and have better progression-free survival. What was also interesting in the study was that they demonstrated that, you know what, perhaps ixazomib in patients who might have remained MRD positive, so people who were MRD positive in the beginning and remained MRD positive, perhaps ixazomib actually helped keep a lid on things. And that's very interesting, because are we saying that, you know what, there may be situations in which we can utilize our maintenance drugs, other mechanisms of trying to see if we can keep a lid on the myeloma, even maybe we can push the MRD deeper, maybe we can keep them a lid on the myeloma to the extent that we don't have to be as concerned about relapse, and we can work towards a functional cure, if not a cure. Here at Hopkins, Yvonne Borello and others had a vaccine trial which used bits and pieces of myeloma to inoculate patients who had a relatively well-controlled myeloma, but not in complete remission or near complete remission, with bits and pieces of myeloma and an immunoadjuvant to see if we could get things under better control. And the goal was to try and see basically if we could go from a state where you had a little bit of MRD to a little bit of a deeper response, or at least get better immune surveillance. And we had a phase 1 study which showed very promising results. We had a phase 2 study as well, which unfortunately fell by the wayside in the era of the pandemic. But we have plans to reutilize a therapy that might potentially be non-toxic and leverage immune environments that might be favorable to it to see if we can help patients with myeloma get things under better control. Over time, what we're seeing is that MRD in clinical trials is being used increasingly. And Even today, though, the bulk of trials, they use MRD as an adjunct. It's not necessarily the primary endpoint, except in a couple of trials, for instance, such as Master, Midas. There are others that are in the pipeline. But you'll see at the tail end of the curve that these numbers are beginning to go up. And that we'll find is that going forward, people will use MRD more as a clinical endpoint. But why should we use it more as a clinical endpoint? I think that MRD as a clinical endpoint, especially when it's validated, as it has been across the multiple different trials that I showed, is very useful because with the advent of all the new drugs, and the drugs keep getting better and they keep moving in earlier lines, there may be many, many years before we read out to a point where a new drug might potentially be able to give us an answer on progression-free duration or overall survival. Now, those are the gold standards, and nothing argues with that sort of success. Nothing argues with success like success. But there are increasing data that show that, look, you can use MRD negativity as an endpoint into itself, and that will help inform how this treatment behaves, what its implications are for patients in different risk settings, and that can also help make a decision as to drug approvals, because we don't want patients to necessarily wait a long time for a drug that might not be particularly toxic, but maybe potentially produce responses that may well be sustained, especially if MRD negativity as an endpoint shows those benefits. And one has to think of frameworks for doing this, which our European regulatory agencies and the FDA have been working on. Here in the US, we're looking at a framework for trying to use MRD as a primary endpoint. It was built into the master study, and there will be others coming into the pipeline. But I don't think this is something that we can do without. I think finding better ways to find residual myeloma and find what implications that might have and learn from that is really very important for us. And so essentially, just some conclusions. Look, MRD is promising. It certainly is. It's perhaps one pathway towards a cure or a functional cure, but it's not a cure in itself as of yet. And when I see a certain degree of MRD positivity, I always try and look at that in context. The big picture always matters, and imaging matters, disease risk matters, where we are in our treatment process. Are we early? Are we later? All of these things matter. High-risk disease still has some implications in the MRD setting, but there are studies that show that MRD negativity in high-risk disease does improve outcomes. Sustained MRD, this is important. Sustained trends are what matter. And then MRD, we need more trials, prospective trials with MRD itself as an endpoint. And we have to think of clever ways to try and address a point where we get patients to a well-controlled state where they're still MRD positive. How do we come up with new strategies to keep a lid on things? And then, of course, intrinsic factors with testing, sensitivity and standardization do matter. I personally think that if we're going to be going forward, then with the endpoints of MRD endpoints in clinical trials, then there's a trend towards using NGS, especially here in the States, which I think is a very encouraging trend, because that helps give you it's standardized across the institutions and trials. And I think with that, I'll stop there and answer some questions. Thank you all very much. Thank you to our collaborators, our supporters, and especially our patients and their families. Thank you.
Thank you, Dr. Ali. That was great. I really appreciate how much depth you went into it, and especially the case studies that helps us understand how it really works in the real world. Clinical trials are one thing, but the case studies show us how much goes into really considering for each patient what's appropriate. Well, there's lots of questions here, and I've tried to group them accordingly. I'm excited to start the conversation with you. I think we're going to start with fallacies in the MRD, or things that could potentially, where it might be lacking. So as we all know, MRD is generally used using bone marrow biopsy samples, and we'll get into other methods using blood later. But it is possible, is it not, that bone marrow biopsy could have missed a sample of myeloma cells?
Yeah, so thank you. That's an excellent question. So, it's these are thoughts that go through my head and our patients every time I see something. If I see a situation where someone has a well-controlled myeloma, they're doing well on their, let's say, Revlimid maintenance after transplant, and we did a test and it was MRD negative, and we do one a year later, and now it's MRD positive in a minuscule fashion. So the thing I need to sit down and decide is that, look, is this real? And is it really changing? Or is it the fact that had we gone an inch to the left, it would have been different? And I think all of these kind of have implications. So when I see one test and I see that it's positive, I really, in med school and in residency training, when you see a blood test that looks completely out of whack, the first thing they tell you is, why don't you repeat the test? And so I look at what the test results are. I look at where my patient is. Is this well controlled? Does this patient have a history of myeloma that was very easy to control and has stayed controlled for many years? Does this patient have a history of a myeloma that has traditionally grown very rapidly? Do I need to repeat this MRD in 3 months? Can I do it in 6 months? Do I have a PET scan? Because this is a patient that, in fact, has, I can't trust the labs because they don't make a lot of proteins. And I really need to look into the marrow and the PET. So this is absolutely true. There are a lot of fallacies. They can change over time. Getting samples, and nobody likes it. Nobody likes recreational bone marrow biopsies, and but getting additional samples is helpful. With I think there was a question about, and you had mentioned, can we move this into the blood space? I think being able to reliably just use blood testing to show very low levels of myeloma will happen one day, but we're not there yet. I very much hope so for my patient's sake. But right now, we need to go to where, unfortunately, the root of the problem is, which is the marrow space.
Yes, thank you. Another question, there can be multiple clones in a patient with multiple myeloma. If NGS, or this next generation sequencing, is based on initial clone DNA, how can it measure later dominant clones?
Yeah, so that's a good question. It's a great question. It's a very technical question. So we know that there is clonal heterogeneity over time, and that sort of makes sense. What you're saying is that there is this here cancer cell, plasma cell, it went bad, became cancerous. Something happened to the DNA, and now it's multiplying willy-nilly. And each time it multiplies, and there's a new generation of kids or myeloma cells, they're different from the last cell, but they have certain traits that they retain and that do continue. And that's kind of inherent to the biology of VDJ rearrangement that happens before the plasma cell fully becomes a plasma cell. So a lot of those are retained. So even if you look at clones that come much later, and when you do the next gen sequencing, they look for a lot of the adaptive test is looking specifically for the VDJ sequence, but next gen sequencing can look at the whole genome of the plasma cell. And it can tell you over time that, hey, look, there's a lot of things that have changed, but you'll find that a big chunk of the VDJ is retained. But that doesn't take away part of this question here, which is that, look, if we're retaining some of the initial clone, right? Because patients do start out as MGUS, and then over time, something happens, right? So, and something changes. So maybe what happens is that you beat your myeloma back to a point where what's left is a non-dominant clone. The clonoSEQ adaptive test is still able to identify those and tell you whether this is part of the original clone that it looked at, because that's what it's looking for. It's looking for VDJ, which to a large extent is conserved over time. Additional next-gen sequencing and directed sequencing can look at how different are these clones from earlier clones. What is their immune profile? Are these going to behave aggressively? Are these not going to behave aggressively? A lot of this is research, and research built into trials and research built into institutions. But what we want to do is be able to operationalize this for everybody, right? So that we can tell a person, I can tell my patient with a great degree of confidence that, look, when you pop a lambda light chain on the immunofixation, I am not concerned at all. Right now, what did we do each time that it came up? I was like, hey, look, why don't we just see where things are in a few months, which is what we've done. And for our success is a good sort of indicator of future success in some contexts as well. But what I'd like to be able to do is tell patients through a non-invasive test, like a blood test, with perhaps some imaging and some other tests that don't really involve being stabbed in the back, and say, hey, look, we've taken a good look at the DNA and the immune profiles in your cell. We've taken a good look at you and your immune profile and your microenvironment. And we would like to be able to say with a reasonable degree of confidence, this is how things will behave in the future.
Yeah.
We're not quite there yet. That's somewhat crystal ball, but hopefully in the years to come, absolutely.
Yeah, I admire your confidence and thanks for educating us on that. Following the NGS discussion, Andrew's wondering, can patients who have had myeloma for many years and have not had a recent bone marrow biopsy get next generation sequencing since there's not a recent baseline sample?
So that's a great question. I have a gentleman who was diagnosed with his myeloma back in 2010 or 2011. And we tried to get adaptive next-gen sequencing by the adaptive test, and they simply could not find or get a hold of the sample from the 2011. He does not have a particularly aggressive myeloma, and I do follow his MRD, but what I convinced him to do was to use a later sample. And if they don't have an initial sample, the very first sample, they can follow samples that have enough myeloma from later. That can be done. But and again, VDJ is retained, but it's not ideal. What is ideal is to have a very first sample where it was all untouched, where you probably had maximum myeloma or someone had maximum myeloma, and say, hey, look, I've got this original sample. I can always compare everything else to my original. So I do follow patients. I have a very small handful that where we used a sample as a for clonality that was later, but there are many that where we cannot get the first sample. Where it becomes an issue, for instance, is that if I have a patient who's had transplant and is now in complete remission, then we try and do MRD negativity, and they cannot get the very first sample, well, then there is nothing in between for me to use to be able to say that, well, I have something else to compare to. So we unfortunately have to wait till there's enough myeloma, which is not something we want for any.
Yeah, thank you. So I'm going to switch gears to talking about minimal residual disease and how it relates to a cure and identifying patients that have been cured. And I know there's so much that we don't know in this space, but I want to communicate the questions that I get frequently and that our audience has. So would you consider MRD negativity as a good sign to help us know which patients are cured?
Yeah, no, that's an excellent question. And I think that question has been around in one form or another since MRD negativity showed up. And I think, as I said earlier, MRD negativity is one tool to assess cure. And human beings cannot necessarily be reduced to a 3-letter acronym, which is a surrogate for a number. So we are complex beings. And MRD negativity is certainly one aspect of us, but there is a lot more to it than that. So I think MRD negativity as a central part of demonstrating cure will be important, right? It certainly will. We'll have MRD negativity, hopefully better tests. We'll have scans, hopefully even better scans. And we'll have patient environments and immune profiling tests that we can do all the time. But now, sometimes, right, as many patients are aware, is that, look, doc, I'm in a VGPR. There's still proteins there, but the MRD was negative. But does that mean that the sample went into a dry patch? Or does that mean that these are proteins that are lingering? Or maybe it's a post-transplant MCUS, that can happen. Or it's because I may not have the best kidneys, and so these proteins, when they're really teensy-weensy, the kidney has trouble picking them up and dumping them out. Which of the above is it? And those are difficult determinations to make. And so I don't try not to do it based on one sample. I do try and get serial samples, and I do try and look at the whole picture. I do think MRD and future tests that are the new MRDs will be a core part of us being able to tell someone that, hey, that look, we think you're cured. And but I don't think it's the only thing.
Thank you. I appreciate your insight. Going along with that, do you believe that there are patients out there who have been cured? And do we have any way yet of identifying them, or do you see a way in the future?
Yeah, I believe that there are patients out there that have been cured. I do believe that. I believe that when you use the stringent definition of cure. I also believe that when you're a little more lax with your definition of cure.
Yeah.
There was another patient who's I didn't put in there who had a large plasmacytoma back in the early 90s, 1990, big one, had to get surgery and radiation to have it cut out. And um, I forget, there was a relapse somewhere in there. He received some treatment or did not. This was in the early 2000s, perhaps. And every year he comes in once a year. He's living his best life, and he's got an M spike, an M protein. So is he really cured of his plasma cell disorder? But maybe he's cured of the myeloma. Actually, he is, right? He's not in any treatment. So what if he has a little bit of an MGUS, so long as this entity stays where it is? So I think between the intensive treatments that we have done for a lot of patients that have gotten them into a real deep remission, where by best testing, we have not found anything, and these patients that skirt the edge of what we consider to be a strict cure, I think there are a lot of people that would qualify for cure and functional cure. And I think it's a little bit difficult to identify. If I have a 45-year-old and I hope for them to make it to 99 and die in a skydiving accident at the age of 99, then am I going to be able to, say, if they ask me at the age of 50, am I cured? Well, I'd like to say yes. But really, in my mind, I'd like to say that you're going to get to your intended lifespan, and maybe there's no myeloma, or maybe all you're doing for your myeloma is taking a shot every 6 months, or maybe a pill once a day, and you're living your life climbing mountains. So those are all, I think, achievable and viable entities. I hope to be able to get to a point in time where you can just walk into your doctor's office and get some cells and some aspirin, and then you're done for the rest of your life. And I think we'll get there. I don't think it's science fiction. And I think cellular engineering and CAR T cells and all MRD, these are all in their infancy. So it will happen one day. We'll get to Mars one day. We'll cure myeloma one day, and it will happen. It's a process, and we're well on our way. And I think everyone here is interested and supportive, and that's the goal.
Yeah, I agree. Okay, let's switch back to the blood-based MRD testing with the few minutes that we have left. Can you briefly explain mass spectrometry and what they're trying to do with this test?
Yeah, no, thank you. That's a great question. I left the mass spec bit out because I wondered if it might be too much for what we were trying to talk about. So mass spectrometry is an excellent test, and a lot of people are using it to look for myeloma proteins. And what mass spectrometry does is, in a very sensitive fashion, looks for the presence of any kind of protein. You can kind of set it to look for whatever protein you want of a certain size and shape and charge. And and it's very, very sensitive. And our colleagues at the Mayo have shown in various studies, in fact, as well as others, in time of flight mass spectrometry testing, that if you look at patients who may have myeloma now, and if you have some, because they have these longitudinal samples, they go back and look at samples from earlier, and they look to see if there was an SPEP or an immunofixation, and they see that all of that was negative. But they do this very sensitive test called using mass spectrometry. And they find this protein, and they're like, you know what, there was some evidence of MGUS right at the beginning. That's very interesting. That's very helpful. Well, if we can find it when technically there was no MGUS or myeloma, then maybe these tests can be used afterwards as well. And I think it will continue to increase in use and in validation. But I think there have to be more studies done that can we use this and do away with stuff like SPEP and so on. And I think SPEP and so on will probably eventually go away, not for the next few years, but probably after that. And part of this is that they're not as sensitive as tests like mass spectrometry. If there's anything about mass spectrometry that's interesting, is that potentially it's very, very sensitive. And that matters less after you're already being treated for myeloma. But if you're being screened and they find a protein that may actually have no implication at any point in time in the future, then that raises certain questions, right? Because I have high cholesterol, I have a PCP appointment on next Friday, and I'm afraid of what she might say. I have trouble with that anxiety. What if we have to tell patients, hey, look, we have this newfangled test, and I know you're 24, but we found this teensy weensy protein in there. Well, what are the implications? Do I have to live with this for the rest of my life? So before we inflict all these things on patients, we really have to test them and validate them. But it's a good and a sensitive test, and we do like it, and you'll find that over time it has more and more utility in conjunction with the MRD tests and other tests that we do for myeloma.
Yeah, really good point, because it's not going to be a one test that we rely on, but rather a series of tests to make sure that we have the complete picture. I
Absolutely.
appreciate that insight. Well, I'll finish with this one question here. Actually, a couple mini questions. So
Mhm.
Derek was wondering if you could define microenvironment in the context you were talking in the clinical trials. You mentioned microenvironment. Do you mind defining that a little more?
Yeah. So plasma cells, it's a strange sort of thing between pathologists and oncologists. Oncologists will tell you, hey, I've got a patient with myeloma, which is a plasma cell disorder. I have trouble killing the myeloma and plasma cells inside the patient. The moment you take them out, though, and you send them over to the pathologist is like, yeah, you know what? I can't use this sample. The myeloma cells are already dead because they're really fragile outside the bone marrow. Why is that? Is that the bone marrow has a lot of other cells. Those are immune cells, those are support cells. It has other molecules and chemicals and a background and a matrix that cancer cells co-opt to remain alive. And changes in the tumor microenvironment that include what are the immune cells like? What are the other supportive cells like? For instance, the ones that deal with bone health, because myeloma changes that. Myeloma patients, as we all know, can have a lot of bone issues. And all of that is part of what the interplay of the microenvironment is. There have been a lot of studies looking at what the implications of the microenvironment are for myeloma. There are some signals about, say, a healthy, robust microenvironment that makes it easier to control myeloma. There are some signals that show that when myeloma is aggressive, it suppresses a lot of things, does it immune suppression, and co-opts the microenvironment. And we know this is the case. We are growing our data about it. We don't quite know how to change it. One example, actually, that I will give you is that Dr. Brello, when he was here, had a test or a trial of marrow infiltrating lymphocytes. And what that means is that he went into patients' bone marrows who had high risk disease, and he took samples of immune cells from the bone marrow. And his thought process was that, look, these cells are exposed to the cancer. They recognize the cancer, but the cancer produces molecules and co-opts the microenvironment to suppress these cells. So to suppress these immune cells. So if I take them out and then reinsert them after taking away the suppressive immune effect and put them back in there with drugs that might help boost the immune system, Revlimid, other drugs as well that he had in mind, then the whole process was to kind of manipulate the immune microenvironment of the bone marrow to make it favorable to recognizing and killing myeloma. And there are many people, many researchers out there that are looking at ways to leverage that part of the tumor, the bone marrow microenvironment and beyond.
Thank you. Another definition question, sustained MRD negativity. Would you consider that to be 2 consecutive MRD negative tests or 3? When do you count it as sustained?
Yeah, that's also an excellent question. So there's the IMWG way of looking at it, right? Which is that, look, if you want to declare a change, it has to meet the criteria and it has to be over 2 contiguous time points, right? But those criteria, those are, for progression really are fairly large, right? And M spike has to be more than 0.5. What if somebody comes to me and says, doc, you know what, I'm on maintenance, I still have an M spike of 0.1. Now it's 0.2 or it's 0.3. So for me, multiple serial time points need to show that, look, that's a significant change. Assuming that these proteins can be followed. Again, there are patients where proteins were small to begin with, maybe not as reliable. But if the proteins have been reliable, or I already have good imaging in a marrow, then what I want to look at before I subject someone to, hey, let's change everything, I want to make sure that, look, this is not something that is a blip that's going to go away, because we see that happen. I see patients where the numbers edge their way up and then edge their way back down. I see this, and I tell patients, it's okay if I see this. What I don't want to see is this.
Mm-hmm.
Sometimes you have to, it takes time. you have to kind of get comfortable playing chicken with the proteins and knowing when to flinch, not too soon, not too early. So that's a sort of a collaborative thing, and you have to watch closely. Again, labs, marrow if it's going to help you see what's going on, imaging if it's going to help you see what's going on, and a lot of reassurance.
Thank you. Well, Dr. Ali, thank you so much for an excellent session full of education. MRD is such a complicated subject, and I feel like you gave us a lot of great information today to be able to clarify some things and think about other things that are coming in the future. Are there any closing remarks that you'd like to make before we finish up for today?
Thank you guys so much. I find you guys very, very inspiring. Those of you who are my patients and those of you who are not. This is a team effort, and nobody, not especially not the myeloma experts, know everything. So we always sit down. Every time there's an opportunity for us to sit down and see, hey, what can we do to make things better for our patients? Please talk to your doctors. They are collaborative. They're your colleagues as well. You're the part of your team in helping you get through this myeloma journey, and it is a journey. But thank you all so much.
Thank you very much. We appreciate you.
Thank you.
To my audience, we're just going to close up with a couple of outro announcements, and then I'll let you go. I want to be respectful of your time. We do have a couple upcoming events next week. June 27th, we're going to be talking about nutrition and wellness for myeloma and sugar and cancer. There's so much out there. Some of it's true, some of it's untrue about the correlation between sugar and cancer. I know some of you are significantly worried about this topic, and so I wanted to make sure that we talk about it. So that's going to be on the 27th at 1 p.m. Eastern. And then that same day at 6:30 p.m. Eastern is our Florida myeloma community chapter. We're going to be talking about understanding your myeloma labs. And then on Wednesday, June 28th, which is my birthday, nobody asked, but I just wanted to tell you, we're going to be having our Spanish chapter. So if you or your loved one speak Spanish and would like to attend, we're going to be talking about mental health and how we can properly manage that. The link to sign up for any of those events and even more events I didn't mention is found at the bottom of the slide and will be included in our follow-up email. Another thank you to our sponsors, Amgen, AbbVie, Adaptive Biotechnologies, Janssen Oncology, Genentech, and Bristol Myers Squibb. Thank you all for being here, and I hope you have a great rest of your day. Take care, everyone. Bye-bye.